Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get into med school and other professional programs. Today, we're going to continue our amino acid learning by focusing in on exactly what you need to know about how amino acids work for the MCAT. We will look at several different topics in this video, such as intermolecular forces between R groups, which amino acids covalently bond using their R groups, where polar amino acids are likely to be found in a protein, and finally, where nonpolar amino acids are likely to be found in a protein. Now let's jump right on into it. Firstly, let's discuss intermolecular forces between R groups. The R group, also known as the side chain, is what differentiates one amino acid from the other. It is responsible for the unique chemical and physical properties of each amino acid. Depending on the nature of the R group, amino acids can form different types of intermolecular forces. For example, polar R groups can form hydrogen bonding, seen here between these two serines. We see that we have hydrogen bonding going on between their R groups. However, hydrogen bonds aren't the only type of R group interactions we'll see. If you look over here at I with their isoleucine, these are two very non-polar groups, but you can see there's still going to be a force of attraction between them. This is due to van der Waals forces. These interactions between R groups can significantly impact the protein structure as well as function. The MCAT will frequently test your knowledge of the individual amino acids by asking you to figure out the overall charge of a small peptide at a certain pH. In addition to understanding the intermolecular forces you are likely to see, you should also know the charge of all of those R groups. So as a quick refresher, our basic amino acids are going to have a +1 charge, and our acidic amino acids are going to have a -1 charge. Everything else is going to have a flat 0 charge. This is important to know because the MCAT will frequently test your knowledge of these charges of individual amino acids by asking you to figure out the overall charge of a small peptide at a certain pH. Another way this is commonly tested is through asking you something about SDS. Now, SDS is used to coat all amino acids in a negative charge, so we can run them on gel electrophoresis and have a nice even charge coating. So to recap what you know about intermolecular forces, polar amino acids are likely to interact with each other through hydrogen bonding, and nonpolar amino acids are likely to interact through van der Waals forces, and SDS can be used to coat all amino acids with a negative charge. Moving on, let's discuss covalent bonding between amino acids. While peptide bonds connect amino acids together on the backbone, as highlighted here in red, one amino acid can also form covalent bonds with another one via their R groups. This amino acid is cysteine. Cysteine has a sulfhydryl SH group in its R group that can form a stable disulfide bond with another cysteine molecule. These disulfide bonds are crucial for stabilizing protein tertiary and quaternary structures as well as their function. A common way the MCAT asks you about these covalent bonds will be in the context of a reducing agent or technique, like a reducing SDS page. When a protein is subject to these conditions, we'll see that the bonds between the sulfurs are going to break, and we're going to stick on two hydrogens again. And these amino acids are no longer covalently linked, and as you can imagine, this is going to have dramatic effects on the protein structure as well as function, usually in the way of reducing that. Next, let's talk about the location of polar amino acids in proteins. Polar amino acids are hydrophilic, meaning they will have an affinity for water. Consequently, they tend to be located on the surface of proteins, or anywhere that can interact with water molecules. That includes the inside lining a channel across the membrane. Polar amino acids also tend to form hydrogen bonds with one another, which can contribute to the stability of protein structure. Lastly, let's discuss the location of nonpolar amino acids in proteins. Nonpolar amino acids are hydrophobic, meaning they have a low affinity for water. As a result, they tend to be located on the interior of proteins away from water molecules. Nonpolar amino acids can interact with one another via van der Waals forces, which can help stabilize the protein structure. Finally, to sum things up, amino acids are incredibly versatile molecules that play a crucial role in the protein structure and function. By understanding the intermolecular forces between R groups, the covalent bonding between amino acids, and the location of polar and nonpolar amino acids in proteins, you'll be well equipped to tackle any questions about amino acids that come your way on the MCAT. And let me tell you, there will be a lot of amino acid questions coming your way. Thank you so much for watching our second video on amino acids, and I'll see you next time.